evening to Dr. Shabinas and my fellow classmates. We are from Group 8 and today we would like to present our protein, which is human serum albumin. So before we go more into our presentation, I would like to introduce my teammates. I am Angeline and we have Danuska, Divyasri, Husna, Muhammad Aslam Khan and also Yalni in our group. The word albumin derives from the Latin albus, which is white. It dates back to the recognition that the albumin of an egg, which appears white after coagulation and is a large protein in nature. Today, albumin is generally regarded to mean serum albumin or plasma albumin, the single protein species which is the subject of this article. HSA is a type of, next slide. HSA is a type of transport protein and it comes from the blood plasma of humans. HSA structure is composed of three domains which are homologous in structural features. The structure includes domains 1, domain 2, and domain 3. HSA is a single chain protein with a low molecular weight which is 6,600 kilodalton, which contains 585 amino acids. HSA is a multifunctional, non-glycosylated, negatively charged extracellular plasma protein. Its isoelectric point is 4.8 and it is soluble best at pH 6 to 7. It has a high temperature coefficient of solubility with the optimum temperature of HSA is 62 degrees Celsius. HSA synthesis occurs in a hepatocyte cells but isn't stored by the liver. It is released by the liver, the most abundant protein in human blood plasma. HSA has psychological effects on human metabolism level and it is known as a multifunctional plasma protein. Its function as a carrier of protein, hormones, vitamin, drugs, which includes anesthetic drugs. Serum albumin is a valuable biomarker for liver function synthesis as well as inflammatory disorders, brain tumors, cardiovascular risk disease, and so on. Thus, human serum albumin is chosen as the protein of interest because of its multifunctional availability, biodegradability, and lack of toxicity, including the low cost. The extraction of protein is a challenging process that often requires management for precision. The method of choice depends on how fragile the protein is and how sturdy the cells are. The source of protein of interest used in this analysis can be obtained from the human blood plasma. The abundant albumin that is present inside the human blood plasma accounts for 60% of the plasma protein. There are other protein sources where the albumin can be obtained such as eggs, beef, and fish. However, these sources do not provide the necessary volume of albumin needed because of the low albumin count and it also costs more extract albumin from these sources of protein. Therefore, extraction of albumin from the human blood plasma is the best source for the extraction of albumin. There are various ways to extract albumin from these protein sources, which are the Korn method with combined chromatography, the Korn method with liquid chromatography, purification from placenta, and so on. Each of these presents their own way of extracting albumin, and they also come with their own advantages as well as disadvantages. Upon careful consideration and decision making, the method that will be used in this analysis would be the Korn method liquid chromatography. The method is based on the Korn method where it uses ethyl alcohol to fractionate the plasma protein until the desired protein is obtained, which is albumin. However, this method comes from its disadvantages, which are that the purity of this albumin produced is not high, the protein is easily denatured and requires a cold environment for the reaction to take place. Therefore, with the addition of liquid chromatography, it surpasses the flaws that was mentioned previously, resulting in a great percentage yield of albumin while using low-cost method to achieve it. The extraction process of human serum albumin begins with the use of frozen plasma that undergoes centrifugation, resulting in the removal of chiral precipitate and the factor which will later be used to synthesize other materials. Next, the centrifugation of chiral supernatant, which differs from the previous centrifugation as ethyl alcohol introduced with the chiral supernatant, and the after product are globular and other plasma protein that are removed to produce a higher amount of albumin in supernatant form, which is then used with the liquid chromatography at pH 5.2 to ensure that the presence of albumin only in the plasma due to a low pH it exhibits. The albumin is then undergoes through a heat treatment where it becomes albumin-free fatty acids, a crucial step as it removes the water content present in the plasma and it prepares the albumin for the second chromatography. Sulfacryl S200HR is used in the second chromatography to remove all the remaining globulins, polymers, and protein aggregates. This extraction uses two different chromatography so that the albumin obtained will be high quality with pyrogen-free products which reduce viruses inside it and guarantee the biological safety of the albumin produced. The monomeric albumin is then sterilized by the filtration to reduce the viral inactivation for the safety use of it. As a result, a concentration of 20% albumin extract can be obtained via Korn method liquid chromatography. Now, I would like to pass the presentation to Husna to present further on the purification process of our protein of interest.
Thank you, Angeline. I will be continuing this presentation with the purification of our protein. Since the extraction of human serum albumin from the blood plasma was completed with the resulting concentration of 20% albumin extract, the next step conducted was purification of the selected protein with the purpose of obtaining purified protein for further accurate protein analysis. The purification method chosen for the separation for albumin from vital foods is the ion exchange chromatography (IEC). IEC has its advantages such as the ability to distinguish protein that differ by just one amino acid. Besides that, ion exchange has only one interaction involved in the separation, which is the analytical species interacting with the stationary phase. The principle behind this chosen chromatography technique involves the separation of ionizable molecules based on their total charge. This technique is commonly used to separate charged biological molecules such as protein, peptide, amino acid, or nucleotides. This technique also enables the separation of similar types of molecules that would be difficult to separate by other techniques, because the charge carried by the molecule of the interest can be readily manipulated by changing the buffer pH. In order to accomplish this separation, the pH of buffer used must be precise in order to separate the protein of interest, as it determines the net charge of the protein of interest. The principle behind this is that in a buffer with a pH greater than the pI of the protein of interest, the protein will carry a net negative charge. Therefore, a positively charged anion exchange resin is chosen to capture this protein. The buffer used for the solution preparation was formic acid buffer, which has a 50 molar concentration, a pH of 3.6, and sodium ion as its counter ion. As the pH of the buffer must be 0.5 to 1.5 units below the pI of the protein of interest, and the pI of interest is 4.8, therefore the pH of the CM cellulose resin should be below 3.8 to act as a negatively charged cation exchange resin. Summary of technique of albumin purification. After the preparation of the buffer is done, resin was washed 3 times with the primary buffer volume. Then the sub-buffer was added to the resin, followed by plasma. The mixture was shaken for 1 minute at 2,500 rpm in a shaker and centrifuge at 1,300 rpm for 5 minutes. This is to obtain the yielding supernatant, and the rest was wasted. The start buffer was added to the solution obtained just now and stirred for 5 minutes at 2,500 rpm in a shaker and centrifuge at 1,300 rpm for 5 minutes. This last solution was combined with the starting buffer into the vial number 1. This process then repeated for vial number 2 and number 3 using the same method. The process of shaking and centrifugation were repeated twice to obtain accurate results in data. Finally, the data were examined using STH procedure using 60 volt current for 3 hours. STH is an electrophoretic system used for separating the proteins on the basis of their molecular weight. The principle behind this technique is that a charged molecule migrates to the electrode with opposite sign when placed in an electric field. The rate of migration also influences the structure and charge of the protein. The advantages and disadvantages of SPH. SPH enables the determination of the molecular weight of protein in a sample and also the detection of specific protein on the basis of the character and mass. However, it is not helpful in identifying the DNA or RNA structure of a strain. As a result, pure protein of interest will be successfully obtained from the purification method used with more than 90% of albumin. I would now like to pass the presentation to Yalini to present further on the amino acid analysis of our protein of interest.
Thank you, Husna. Now, I'll be explaining the amino acid analysis of our human serum albumin protein. Amino acid sequencing is the process of identifying the arrangement of amino acids in protein and peptides. Amino acid sequencing. The amino acid sequence of a protein can be identified by the Edman degradation process, which was introduced by per admin. This is a process where the protein will be purified by removing one residue at a time from the amino end of the peptide without disrupting the peptide bonds between each amino acid. This technique is really beneficial as it's sensitive, simple, inexpensive, and does not damage the protein. And it's also done in less time. However, the downside of it is its lack high throughput capabilities as sequencing proceeds on sample of single proteins only. Protein sequencing is really important to determine the amino acid sequence as well as the conformation of the protein. In this sequencing technique, the human serum albumin will be used to study the protein sequencing by the Edman degradation process. Now, let us see how the human serum albumin is being sequenced by Edman degradation technique. Initially, the monomer of human serum albumin will be prepared as starting material. The N-terminal of the human serum albumin is to be determined. Edman's thiohydroton method will be applied to the protein of interest, which is HSA. 3 fragments of HSA will be isolated and the N and C terminal of the amino acid sequence will be labeled. The phenyl isothiocyanate PTC derivatives of protein will be prepared. The HSA will be dissolved and mixed with many chemical materials. And after being stirred continuously for 1 to 2 hours, the reaction mixture will be extracted 6 times with benzene. The residual suspension will be evaporated to dryness in vacuum and washed in acetone and ether. Acetic acid hydrogen chloride will be used for cyclization of PTC derivatives, followed by the dioxine hydrogen chloride, formic acid, and also citrate buffer. The phenyl thiohydroton PTH derivatives will be extracted following the cleaving process. PTH derivatives will be identified by the paper chromatography. After being shaken for 3 hours, the mixture will be extracted thrice with the ether to remove unchanged 1,2,4-fluorodinitrobenzene FDNB. Then, the dinitrophenyl DNP amino acids will be identified in human serum albumin. Then the DNP proteins will undergo acid hydrolysis, and aspartyl-alanine will be identified as the human serum albumin's N-terminal sequence. So, this is how the human serum albumin is actually sequenced before further used. Now, let us see what are the applications of human serum albumin. HSA serves as a drug delivery vehicle due to its characteristics like long half-life, recirculation, and also accumulation in tumor tissues. HSA acts as a carrier protein for steroid, fatty acids, and thyroid hormones, and also plays an important role in stabilizing extracellular fluid volume, and also widely used clinically to treat serious burn injuries and also hemorrhagic shock. For your information, the market demand for HSA is estimated at more than 500 tons per year worldwide. Currently, the commercial production of HSA is primarily based on collected human plasma, which is limited in supply but really high in clinical demand. It was reported that the shortage of human plasma led to a rapid increase in price of HSA, which in turn resulted in fake albumin appearing on the market. Now, I'll pass it to Angeline to conclude our presentation for today on the protein analysis of human serum albumin.
To summarize, human serum albumin is synthesized by the liver and is a transport protein with a molecular weight of 6,600 kilodalton, and it is soluble best at pH 6 to 7. Its isoelectric point is 4.8 and it is a negatively charged protein whereby its synthesis occurs in red blood plasma. HSA uses corn liquid chromatography method and ion exchange chromatography whereby the protein sequencing is through Edman degradation method. HSA helps treat burn injuries, hemorrhagic shock, hyperproteinemia, and helps produce new vaccines as well as pharmaceutical drugs. It is known as drug delivery vehicles and it is also a carrier protein. The following slide shows the references that was needed to produce the slides. And that's all from us today. Thank you so much.